question, besides the one that asks for your student ID number. So what cross do you use to make a dihybrid? Let's see how everybody did. So we've got 3 different options up there. Looks like we don't have to cover that topic. So 50% say choice A, 50% say choice B. Did anybody choose both of those? Few. Okay, so it's both A and B. I was just curious because I wasn't sure how this website displays those results if you choose 2, how it describes those. So why could it be A and B? What are we looking for in a dihybrid? We just want 2 heterozygous genotypes. And so either of those 2 crosses combine a little a and a big A and a little b and a big B. Choice C, what's missing? The reason that most of you didn't choose option C, there's no little a, right? So there's no way to make a heterozygous genotype by crossing those individuals. Any questions about this one? Let's see. 2 genes separated by 35 centimorgans. This is just a question asking you to convert map units to something else. Okay, so some of you are messing with me, or at least one of you is messing with me. So if it's 35 centimorgans, how many map units is that? They're exactly the same. So a map unit is the same value as a centimorgan. So 35 centimorgans is 35 map units. What would that be if we changed it to recombination fraction? You divide map units or centimorgans by 100 to get, choice A. So those of you that picked choice A, it's related at least. That's recombination fraction, RF. 4 is right out, unless you were rounding for some reason. No 4. Ooh, this is an important one. If you cross 2 P0 genotypes, which ones are the parental and which ones are the recombinant haplotypes? Of course, this is critical because we need to find recombinant haplotypes. Why? What are we going to calculate once we figure out the recombinant haplotypes? So why do we care about measuring recombination? Trying to find linkage. So you tell how far apart genes are on a chromosome, or if they're on separate chromosomes, by calculating how many recombinant haplotypes there are. So if you cross big F little g and little f big G, let's see. Great. So the parental types in this case are the only haplotypes that the parents will produce. Big F's with little g's and little f's with big G's. So that individual has big F's and little g's only in their gametes. And that one, little f's and big G's. So those P0 individuals make those parental haplotypes. Their gametes will guarantee you have those genotypes, haplotypes. So in this case, big F with big G is not a parental type. That must be a recombinant type. That would be recombining the big F and the big G from the parents. Recombinant type. And the same is true for little f and little g. Those would be recombinant types. So this is parental. Oops. Big F with little g is parental, B and C. Recombinant types are A and D. Any questions about this? Concerns, clarifications needed? last question. Ah, we've got a smorgasbord with some honest students guessing. Don't think we have enough information. How many people would like to agree with the I don't have enough information? Okay. Few. Okay, whoa. So let's see, we must not have enough information if there's this much diversity in answers. So how would you go about solving this problem? What do you know about the genotypes from this description of the genotypes? How many genes are we looking at? It's not the trick question. 2, F and G. Where are they located in the genome? There's something in there that tells us whether or not the genes are linked or unlinked. The semicolon. Semicolon means genes are on separate chromosomes. If these genes were on the same chromosome and I knew it, how would I write this same genotype? If 2 genes were on the same physical piece of DNA like this. So there's 2 chromosomes with big F there, little f there, big G there, little g there. Or the genotype, how would you write that genotype? Linked alleles. There's no semicolon. If the semicolon means they're on separate chromosomes, so that would be like that and like this. A big G and a little g on one pair of chromosomes, a big F, little f on another. That's when you use the semicolon. So if you got no semicolon, that looks like that. How do you write that genotype? And 2 alleles are physically together. There's a slash. There's 2 molecules of DNA here. So everything that's on one molecule goes on one side of the slash. And everything on the other chromosome goes on the other side. We've talked about this before. If the genes are linked and we know it, you write the 2 haplotypes, big F with big G, on one side of the slash and the little f with the little g on the other side of the slash. So this is all just about interpreting the nomenclature. If there's a semicolon, I'm telling you that the genes are on separate chromosomes. Down here. So what's the genetic distance between unlinked genes? Asking about 2 different things. Interpret what the semicolon means and then tell me how far apart these genes are. So what is the answer to number 5? They're on 2 different chromosomes, so what's the essential question is, which we're going to talk about in class, which is why I haven't told you the answer yet. I mentioned it last time, I'm going to reinforce it with a little bit more in-class activity this time. What's the genetic distance between unlinked genes? So let's imagine that you have a dihybrid. You've got 2 genes, F and G. So this is a tetrad of chromosomes right before meiosis begins. Let's say they're linked. I'm going to number, or letter rather, the individual sister chromatids just for fun. So somebody tell me which 2 should cross over. A, B, C, or D. Which 2 should be involved in recombination? Okay, someone in the back row. Pick 2 letters, A, B, C, or D. And B and C? Okay. Thank you. So we've got a recombination event happening there. How many recombinant haplotypes are going to be produced from these 4 sister chromatids? So what this means is the chromosomes physically break and switch arms. So you get a big F with a big G from this top chromosome. This next one, we pick up a big F here, and then there's a physical recombination event. That chromosome is broken and recombined, literally, with that part of that sister chromatid. So a big F with a little g. And down here we get the reciprocal. We get a little f, part of that sister chromatid that's now connected up there to the big G. So we get a little f with a big G. And down here, by chance, back row, we didn't involve sister chromatid D in this recombination event, so it has not changed. Little f with little g. So what percentage of the gametes are recombinant? How many have been involved in recombining alleles at different genes? How many have stayed exactly the same? 2 out of 4. So it's almost like it is. That looks like a parental haplotype. That looks like a parental haplotype. And these 2 in the middle have recombined. They're recombinant types. Big F with little g, little f with big G. So 50% recombinant is what you expect, either, which we go through a little bit more today, when genes are at the opposite ends of the same chromosome, or if they're on separate chromosomes. 50 centimorgans. 50% of the gametes are recombinant, or a recombination fraction of 0.5. Multiply that by 100, you get 50 centimorgans, 50 map units. So 50% recombinant gametes equals 50 centimorgans. That's when genes are at the opposite ends of the same chromosome or if they're on separate chromosomes. Here I've diagrammed what happens when genes are the opposite ends of the same chromosome. How do you know this is true for genes on separate chromosomes? What sort of thing would we have to draw then? How would I change what I drew there to represent genes on separate chromosomes? You do it for me. What should I draw? If I want to represent 2 genes on separate chromosomes. Should we do meiosis again? Say we've got a heterozygote, so we've got 2 big Fs, 2 little f's, and the same on a separate chromosome this time for G. What happens at fertilization? We've got 2 chromosomes carrying big G, 2 carrying little g, same for F. How do you represent, what do you draw on this slide here when you want to represent what happens at fertilization? Got gamete haplotypes. Want to make the next generation. What comes next? How many different combinations of those alleles can you make at fertilization?
4.
4. So what are the 4? Thank you. So you can get a big F with a big G. So So you could have a sperm carrying a big F, fertilizing an egg carrying and a big G together. Those could be used to fertilize oocytes. So let's say these are the sperm. So we've got different combinations. We can get a big F with a big G. We can get a big F with a little g. So how many parental types and how many recombinant types? We make all possible combinations of different chromosomes that have big F, big G, little g. Every individual, when they go through meiosis, if you're a heterozygote, makes 50% big F, big G, right? So you get all possible combinations. You get big Fs with big Gs, little f's with big Gs, big F's with little g's, and little f's with little g's. Do all the different combinations of recombining genes on different chromosomes. So you still have the exact same thing as on the previous slide. You have recombinant types produced equally as frequently as you get parental types. Regardless of whether genes are on separate chromosomes or the same chromosome, recombination still happens. When they're on separate chromosomes, the recombination is what happens during meiosis, Mendel's second law. Chromosomes line up across the metaphase plate randomly, so you get all 4 possible combinations of alleles, half of which are parental types, half of which are recombinant. When you're talking about genes on the same chromosome, it's physical recombination, the breaking and rearrangement of chromosomes that happens during meiosis that causes half of the chromosomes to be recombinant and have to be parental. 50 centimorgans. If you ever see something more than 50 centimorgans, something's gone kind of fishy. We'll get there. Tester genotypes. So most of you said, accurately, these are both tester genotypes. Homozygous recessive at all loci. So little letters only. A and D don't fit that because there are capital letters in there. So tester genotypes, homozygous recessive. Questions or concerns about any of these questions? Any content up to this point? Clarifications needed? Before we sally forth into more mapping. Yes, please.
So the tester genotypes can be linked?
They can, yes. So they can be linked, they just have to be homozygous for recessive alleles everywhere we go. Linked or not. Thank you. Did you have same question? Others? Yeah.
What was the answer to number 5?
Number 5. Most of you got this one. Vast majority. P-value is less than 0.05. Observed and expected values are significantly different. So initially was false. Should be significantly different. In last class, we used chi-square test and the analysis of p-values like that to ask whether or not in this example the genes were linked or not. So we found that the p-value in this example was some infinitesimally small, p times 10 to the minus 47. The observed and the expected values are significantly different. So the expectation was linkage, opposite, independent assortment. So if the expected value is not what you observe, then independent assortment is not happening, which means genes are physically linked. So when the observed and the expected values don't match, when the p-value is really small, that tells you genes are linked. It's when the observed and the expected values are same that independent assortment's happening, like it should be, like Mendel would have predicted. It means genes are on separate chromosomes. So simple takeaways. Genetic distance ranges from 0 to 50. if you calculate 50 centimorgan genetic distance, you can't distinguish whether genes are on the same chromosome, but at the farthest possible ends of the same chromosome, or if they're on separate chromosomes. Unfortunately for Mendel, people have used statistics against him. Poor guy. Not that he knows. This happened well after he was dead. But recall, or be aware, if you didn't know this, that Mendel worked on these 7 different traits when he crossed peas in his garden in his abbey monastery. Abbey. Wrong gender. He crossed 7 different pairwise sets of traits to try to figure out what independent assortment was doing. He crossed seed shape with seed color, for example, so he looked at whether or not those traits independently assorted. Were they linked or were they on separate chromosomes? Seed coat color and seed color, seed coat color and pod color. So he'd pick any 2 traits. And when he crossed those 2 traits, he always saw that 9:3 to 3:1 ratio that suggested that those genes were on separate chromosomes. But Mendel was almost too good. He worked on these 7 different traits and pairwise combinations of them. Every time, as far as we know, every time he did this, he saw that 9:3 to 3:1 ratio. So if he saw a 9:3 to 3:1 ratio, what would the chi-square value be? If he did that chi-square test to ask if the genes were independently assorting or not. If you get a big chi-square value, what does that mean about the p-value? They're connected. So a very large chi-square value leads to a very small p-value. If you go back and look at the chi-square table. What happens when it's a small p-value? That means the observed and the expected values match or don't. Small p-value, p less than 0.05, observed is equal to or not equal to expected? It's not equal to. So what Mendel saw was what he thinks he predicted. So if his observed and expected values matched, he thought he was going to see a 9:3 to 3:1 ratio, and he actually did when he set up all these crosses. His chi-square value was probably, if observed is going to equal expected, he probably saw large p-values if he did a chi-square test, which means he was looking at small chi-square values. And because he saw what he expected, what did that tell him about where his genes were located? If he observed independent assortment, between all possible combinations of those 7 traits, what does that mean about all the genes that control all of those traits? Are they all linked or are they all unlinked? So when observed equals expected, genes are unlinked, 50 centimorgans apart. Mendel predicted, and we would predict, that all those genes were located on separate chromosomes. So the 7 traits that he studied all act like they're unlinked, 9:3 to 3:1 ratios. You may not believe this. It's maybe kind of true. Mendel's celebrated paper is an inexhaustible source of inspiration and controversy for each succeeding generation of geneticists and historians of genetics, whatever that is. For the aficionado, it is studied repeatedly, and much like an avid sports fan enjoys each rerun of a classic matchup. It's true. Go back and reread Mendel's 1865 papers. Ooh, it's good. But what does this title suggest? Mendel lied to us. The alleged falsification of Mendel's data. Did Mendel actually tell us what he observed, or did he lie to us? So this is a pair of geneticists, Hartle and Fairbanks, who are summarizing some arguments that have been made about whether or not we could use statistics against Mendel to prove that he lied to us. So here's one of the papers that was written, Fisher's criticism of Mendel's results with the garden pea. Basically, this is the point to focus attention on. Mendel's data, actually, if you think about it, what are the probabilities that he happened to choose 7 different traits that happen to be located on 7 different chromosomes? Wouldn't by chance he have happened to have chosen 2 traits that were linked? So what's the chance, this is what they're saying, what's the chance that Mendel actually just by chance happened to pick 7 different traits, pea shape, pea color, seed color, wrinkled versus round pods, all that. And they all happen to be on separate chromosomes. So here's a genetic map of the pea. This is representing each of the different chromosomes. So peas have 7 chromosomes. And the traits that Mendel studied are the ones shown in red. Flower color is controlled by a gene here on chromosome 2. Height of the plant is controlled down here, a gene on chromosome 3. Flower position, seed shape, pod shape. So what's true about all the genes that he studied? Now we know we can actually sequence the genome of the pea. We can find where the genes are located. Are all of the genes really unlinked? Is there one gene controlling each of those traits on each of the 7 chromosomes? Which chromosome doesn't have a gene? 6, yeah. So he didn't have a gene that controlled a trait that was found on the 6th chromosome, but there were 2, pod color and height, that happened to be on the same chromosome. But they're far enough apart that they act, because of recombination, like they're unlinked. What sort of genetic distance would you expect between those 2 genes? What's the genetic distance between a gene there and a gene there on chromosome 3? 50 centimorgans. So we know the genetic length of every chromosome is 50 centimorgans. So in this case, height and pod color, we could guess maybe 30 centimorgans. They're not at the opposite ends of the chromosome. They're somewhat far apart. So I'm just going to say maybe 30. Somewhere in between 0 and 50, clearly. They're far enough apart on the chromosome that when Mendel looked at his data, they were pretty close to independently assorting. So recombination does occur between them frequently. So what do you think? Do you think it's just chance that Mendel happened to pick 7 different traits that are located on 6 different chromosomes? Or did he lie to us? How would he have lied to us? So he is studying unlinked genes. So what might have he done before he wrote the papers that we now study like aficionados of classic sports events? What did he tell us? What did he leave out? Potentially. No one knows if this is true, of course. This is like historical forensics.
Outliers of his data? Outliers?
Sure. So he might have selectively ignored telling us about traits that he studied that happened to be linked because they didn't meet what he was then expecting. He started seeing this 9 to 3 to 3 to 1 ratio. And so every time he crossed 2 traits, he thought, I should see a 9 to 3 to 3 to 1 ratio. And when he saw something that wasn't that, like let's say pod shape is controlled by a gene here on chromosome 7. Let's say there was a trait here that's resistance to drought. The gene that controls how well the plant grows in wet weather versus dry weather. So let's say he made crosses of plants that were resistant to drought and not. He could water them, they were fine, or he could not water them and they were fine. And he never saw those traits being separated. So he never saw a 9 to 3 to 3 to 1 ratio. Maybe he just said, you know what, that doesn't make sense. I'm just not going to record it in my lab notebook. So the argument is maybe he selectively reported his data. But regardless, he did discover the first and the second laws, which are true and accurately represent what happens in genetics. So you can use chi-square analysis to ask these sorts of questions. Did Mendel actually, by chance, is it reasonable, is it likely that he chose these traits at random, or is it more likely that he actually sort of fudged the data? So genes that are 50 centimorgans apart could either be unlinked or at the opposite ends of the same chromosome. One other thing that you need to know to be able to perform really well on the next exam is this point. So we've already covered chi-square test, converting map distances, centimorgans, map units, recombination fraction, identifying parental and recombinant haplotypes. Then we've got this. Genetic distances are additive. So say A and B are 30 centimorgans apart, and B and C are 5 centimorgans apart. That means that, what's the distance between A and C? So what I'd like you to do is, on this slide down at the bottom, draw a physical representation of a chromosome, just a line if you want. Write down the locations. It doesn't matter which order you put them in, maybe. We'll see which order you put them in. Write down a physical map where the 3 genes are located, A, B, and C, that explains how A and B can be 30 centimorgans apart and B and C can be 5 centimorgans apart. Pretend like you're measuring centimeters instead of centimorgans. So draw a genetic map with A, B, and C. And then attach that to the Google Classroom assignment that I posted at the start of class. It's the additive genetic distances assignment. If you have a question, do raise your hand if you want me to come around and discuss. So let's see, I'll just pick a few at random. Thank you all of you for sending these in. Okay, so here are some possibilities. So do these all satisfy the criteria? A and B are 30 centimorgans apart. B and C are 5 centimorgans apart. So you could have the order A, B, C with those distances between those genes. Oh, that's what, by chance, all 3 of these answers look the same. Did anybody do anything different? What did you do? So which order from left to right? Or top to bottom, either way? A, C, B. So how would, yeah, so let's see. How does that work? Right. So if A and C are 25 centimorgans apart, then the distance from A to B is still 30, which was a requirement, and B to C is still 5, which is one of the requirements. The point is that we can't tell just from these data which order the genes are in. So this is another thing that geneticists use recombination mapping to do. It's not just as simple, which is, I know, I've said this a lot, it's not just figuring out whether two genes are on the same chromosome. Beyond that, we can also use genetic distances to figure out the relative order of genes on a chromosome. We're just not there yet. So it could be either of these. Either of these are consistent, either of these maps. For example, either of those three, which are all the same map, and this one, they're all equivalent. We can't tell between them. So here's an example of unknown linkage. So I was mean, I was cruel in the explain everything, or in the Socrative quiz at the start of class. I threw that semicolon in and asked you to tell me that that meant that genes were on separate chromosomes. I'll remind you now that the dot between genes means that we don't know what the linkage relationships are. So this is a type of an example where I'm actually telling you at the beginning, you need to tell me whether or not these genes are on separate chromosomes, or if they're linked. So we set up this cross. What are the P0, here's the P0 generation. What are their haplotypes? What are the parental types going to be in this cross? Big A with big B, little a with little b. What sort of a cross do we have in the F1 generation? The name of the cross, yeah, dihybrid there, tester there, little a's, little b's, homozygous recessive. So what does that mean? How do we figure out whether or not recombination happened when that F1 individual is producing gametes? What can we do in these F2 genotypes? We can cross out the contributions that we know were made from the gamete of the F1, which is little a and little b. So we know that those little a's and those little b's had to come from the F1 tester parent. So how many parental type gametes do we have? If I number these different genotypes, 1, 2, 3, 4, which ones are the parental types? 1 and 2, the big A with the big B, the little a with the little b. That's what we came up with up here. Just by looking at the genotypes of the P0 parents, we figure out the parental haplotypes. So we've got a parental type there, we've got a parental type there. The other two types are recombinants. Little a with big B and big A with little b. So what's the recombination fraction? How far apart physically, map units, centimorgans, recombination fraction? How far apart are these two genes? We take the total number of recombinants over the total number of individuals that we looked at, which happens in this case to be 1,000, to make the math easy, since it's just an example. So what's the genetic distance between A and B? 10, I heard 10 centimorgans. This is 100 divided by 1,000, or 0.1 recombination fraction. Multiply that by 100, 10 centimorgans or 10 map units. Okay, so A and B are 10 centimorgans apart. Are they linked or unlinked? Turn to your neighbor and discuss. 30 seconds. Convince them you're right. Is it linked? Are they linked or unlinked if they're 10 centimorgans apart? Okay, so who managed to convince your neighbor that you're right? Are they linked or are they unlinked? They're linked because? Right, genes have to be 50 centimorgans apart to be unlinked. We expect genes on separate chromosomes to have 50 centimorgan genetic distances. So anything less than 50 centimorgans is evidence that genes are linked. So this is evidence, finding that genetic distance that's less than 50 centimorgans is evidence of genes being on the same chromosome. In other words, if you set up this test cross and you make a Punnett column with the little a, little b gamete from the tester individual there, and the 4 different types of gametes that are produced, presumably produced by the dihybrid, we expect to see a 1-to-1 to 1-to-1 ratio. 25% of the offspring should have each of those genotype combinations. Look up here, or look at your tablet screen. Does that look like a 1-to-1 to 1-to-1 ratio? No way. Which ones are more prevalent? Parental types or recombinant types? Right, there's a vast majority of the F2s are parental types, not recombinants, non-recombinants, non-recombining, closely linked genes. So when you see an excess of parental types, when you see more parental types than you would expect by chance, Punnett square in other words, that's evidence of linkage. That will give you a small genetic map distance. It means you have very few recombinants and a large number of, so the numerator here will be small number of recombinants compared to the total, small number of recombinants compared to the total number of individuals you look at. So A and B, 10 centimorgans. Please do this one on your own. We've got about 3 minutes, maybe 4. Do exactly the same thing. Discuss with a neighbor if you like. And when we come back together, we'll close up, tell you what to do for next class. So go ahead and do this, please. I expect it to be noisy because you should be talking with the neighbor once you're done working this out. Compare answers. In this example, how many F2 organisms do we have? What's our denominator for recombination fraction? It's 1,000 again, yeah. Just trying to keep things easy for calculation. How many recombinants were there?
50.
Okay, so there are 50 recombinants. This is tricky, so I changed things up compared to the first example. The parental type haplotypes are big A with little d. Right, so you have to look, you can't always assume that it's always going to be 2 capital letters and 2 lowercase letters. In this case, the parental haplotypes are little a with big D and big A with little d. So when you get rid of these tester parent genotypes, and you're left with the alleles that were inherited at fertilization from that F1 dihybrid, you've got the big A's with little d's and little a's with big D's, those are the parental types. So hopefully you tallied that the bottom two, 27 and 23, sum to 50. Those are the recombinant types. So what's the genetic distance between A and D? In centimorgans, say.
5.
Yeah, 5 centimorgans. So that's recombination fraction times 100. 0.05 times 100. Now you know the genetic distance between A and B. That's what we did on the previous screen. And now you know the genetic distance between A and D. So 10 centimorgans between A and B, 5 centimorgans between A and D. So I'd like you to do exactly the same thing, same approach that we did when you took the screenshot. I haven't opened this Google Classroom assignment yet. I will immediately. 10 minutes. Do exactly the same thing. Draw a genetic map that explains the relationships in positions on one chromosome or multiple chromosomes, whatever you think is appropriate given our data. Physical map. So where are those 3 genes, A, B, and D, located, based on the 2 mapping examples we just did? When we come back to class next time, we'll talk about new sorts of mapping approaches that will help us answer this question. So office hours from now to 1:30 in case anyone wants to stop by.